Welcome back to the deep dive. For generations, milk was, well, it was just milk, wasn't it?
It really was.
The simplest item in our diet. A uniform white liquid, you drink it for calcium, it comes from a cow. End of story.
That's right. But that view, it's been completely turned on its head by modern science.
We're so far beyond that now. We're not just talking about dairy anymore. We're analyzing it down to the single amino acid, looking at these tiny protein variants and their huge impact on our health.
It's all about proteomics now, the molecular fingerprint of our food. And the core of this whole debate, this A1 versus A2 thing, it really focuses on two versions of the second most abundant protein in cow's milk, beta-casein.
It sounds incredibly technical, beta-casein, like something you'd only hear about in a research lab.
It does, but the reason so many people feel sick after drinking milk, the whole controversy, it rests on one tiny microscopic difference.
Okay, let's unpack that right away. Beta-casein, you said it's a big protein. It's a chain of 209 amino acids, right? Makes up about a third of the protein in milk.
A very substantial part of it, yes.
And you're saying the difference between A1, which can cause problems, and A2, which often doesn't, is just one substitution in that entire chain.
Precisely one. A single amino acid substitution at the 67th position of that chain.
Wow.
In the original, the ancestral A2 version, that spot is held by an amino acid called proline. In the newer A1 variant, it's a histidine. It seems completely negligible.
Like changing one letter in a very long sentence.
That's a perfect analogy. But that one chemical change fundamentally alters the protein structure. It creates a kind of a weak point that dictates exactly how your digestive system is going to process it.
And this weak point, this is the foundation of the BCM7 hypothesis, isn't it? The idea that A1 milk releases something problematic into our gut that A2 milk keeps locked away.
That's it, exactly.
So our mission today is to really do a deep dive into that hypothesis. We need to follow the trail from that single amino acid all the way to its consequences for gut health, inflammation, and maybe even our brain function.
And it's such an important deep dive because millions of people struggle with dairy. They just assume it's lactose intolerance, a sugar problem.
Right.
But the biochemistry is pointing to something much more subtle. It might actually be a protein problem, one that started thousands of years ago with a random genetic mutation.
That sets the perfect stage. To understand the conflict, we have to understand where it came from. So let's trace it back to this evolutionary fork in the dairy road.
Okay. So the scientific consensus is that the A2 variant is the ancestral form. It's the original...
The default setting.
The default setting, exactly. For thousands of years, all domesticated cattle produced milk with this A2 protein. And just as importantly, it's the same beta-casein profile that we see in other mammals, humans, goats, sheep, buffalo, camels. A2 was the global standard.
So if A2 is the dress line, where did A1 come from? How did it show up?
It wasn't gradual. It was a pretty distinct and, in evolutionary terms, a very recent genetic event. We can pinpoint it to roughly 5,000 to 8,000 years ago.
Yeah.
And it happened specifically in European cattle. The Bos-Drossaurus lineages that were being farmed across northern Europe, it was a spontaneous point mutation. A single nucleotide polymorphism, or SNP, on the 6th chromosome.
A point mutation. So literally one letter of the genetic code changed. Can we zoom in on that, on the specific gene?
We can get extremely granular, and it's worth it to see how small the change really was. We're looking at the CSN2 gene, which holds the instructions for building beta-casein.
Right.
At codon 67, the original DNA sequence was CCT, cytosine to thymine. That triplet is the code for the amino acid proline.
Okay, CCT for proline.
The mutation changed that sequence to CAT, cytosine, adenine, thymine, and CCAT codes for histidine. So the entire change that remade the dairy industry, CCT became CT, proline became histidine. That's it.
And this mutation didn't just stay in one small herd of cows, it spread.
It spread like wildfire. And ironically, the very thing that made us so successful at dairy farming industrialization is what accelerated the spread of this A1 gene.
How did that happen? How did human industry end up amplifying this one specific mutation?
Well, once dairy farming moved from small, local farms to these huge industrial operations, the number one factor became volume, milk yields per cow.
Of course. Efficiency.
Exactly. So breeds that were known for being incredibly high-volume producers were favored. They were bred selectively and shipped all over the world. And the poster child for this is the Holstein Friesian.
The iconic black and white cow.
The one everyone pictures.
Mm.
And those breeds, which came from northern Europe where the mutation first happened, just happened to carry a very high frequency of the A1 allele.
Which means the conventional milk you pick up at the supermarket, that generic gallon jug.
It's almost certainly going to have A1 protein in it. Most commercial milk comes from these huge mixed herds. So you're getting milk from cows that are either pure A1 or more commonly a mix of A1 and A2.
So what does that breakdown look like in a typical Holstein herd?
The A2 allele frequency in Holsteins is often somewhere between 35% and 65%. So at best, you're looking at a 50-50 mix in your milk, and often the A1 protein is the dominant one.
But there are still these older heritage breeds that have kept that original A2 profile, aren't there?
They absolutely have, and that's where the contrast is so stark. When you look at heritage cattle, especially breeds from the Channel Islands like the Jersey or the Guernsey,
Okay.
the A2 profile is dominant. A Jersey herd will often have an A2 frequency over 70%, and Guernseys are even purer, often over 95% A2. Brown Swiss, also very high in A2.
What about outside of Europe? What about cattle that weren't part of that same industrial push?
Great question. The Bos Indicus, or Zebu-type cattle, the ones you see in India like the Sahiwal and Gur breeds, they're almost universally 100% pure A2. Same goes for water buffalo, which are a massive source of dairy worldwide, and most native African breeds.
So historically, huge parts of the world were drinking exclusively A2 milk until very, very recently.
That is the critical context, but there's a modern complication we have to mention.
Which?
Crossbreeding. In an effort to boost milk yields in places like India, there's been a big push to crossbreed local A2 cows with high-yield A1 Holsteins.
So we're inadvertently introducing the A1 gene into populations that never had it.
Exactly. It's complicating the global dairy landscape. It means that today, the only way to be 100% sure you're getting pure A2 milk is through genetic testing, not just by looking at the breed or where it's from.
Okay, that brings us to the product in the glass. Let's pivot now to the mechanism. This is where it gets really personal for the listener. Why does having proline at that position 67 create such a strong protein, while histidine creates this weak link that releases BCM7?
It's just a beautiful piece of biochemistry. It's all about the shape, the structural conformation of that protein chain. Proline isn't a standard amino acid. It's technically an amino acid.
What does that mean structurally?
It's unique. Its side chain loops back and bonds to its own nitrogen atom, forming this really rigid five-membered ring.
So it's naturally stiff, it doesn't bend easily.
Precisely. That stiffness restricts the flexibility of the whole protein chain around it. In A2 milk, that stiff proline at position 67 forms an exceptionally strong bond with the amino acid next to it, isoleucine, at position 66.
Okay.
And here's the key. That strong, rigid bond is highly resistant to being broken by our digestive enzymes, specifically an enzyme called pancreatic elastase.
So the proline acts like a piece of molecular rebar. It's reinforcing that spot, defending it from being attacked by our enzymes.
That is a perfect analogy. And because the enzymes can't break the chain at that critical spot, the beta-casein gets broken down into larger, pretty much inert peptides. One is called BCM9, but it has almost no opioid activity, just passes through harmlessly. The strong bond holds.
Okay, so that's the strong A2 version. Now let's contrast that with the A1 variant where histidine is sitting in that spot instead.
Right. Histidine is a standard amino acid. It doesn't have that rigid ring structure. So when you swap out the proline for histidine, you've removed that structural defender. You've created what scientists call a weak link, a preferred spot for cleavage.
So when the A1 protein gets to our gut,
When it encounters those same digestive enzymes, that pancreatic elastase, the weak bond between isoleucine at 66 and histidine at 67 is just easily snipped.
and what exactly is released when that bond is broken?
The cleavage at that weak link releases a very specific 7 amino acid chain. It's called beta-casein 7, BCM7.
And the name casomorphin. That tells you everything, doesn't it?
It really does. It's a potent exogenous opioid peptide.
An opioid? That's a pretty startling word to associate with digesting a glass of milk. What does that mean physiologically?
It means that BCM7 can bind to and activate our mu-opioid receptors. These are the exact same class of receptors that powerful pharmaceutical opioids target.
But its effect is mostly in the gut, not systemic, like a drug would be.
For most healthy adults, yes. The activity is primarily peripheral. We have a huge number of these receptors throughout our gastrointestinal tract. They're like the master regulators of gut function. And we're not talking about a trivial amount being released, are we? The quantitative difference is pretty big. It's massive. This isn't just a trace difference. When you hydrolyze milk in a lab to mimic digestion, the amount of BCM7 that comes from the A1 variant is roughly 4 times higher than the tiny, tiny amounts that might come from A2. That huge difference is what drives all the downstream effects.
We should probably pause here and mention infants because they're a special case, right?
Absolutely. We have to. Infants have immature digestive tracts. Their intestinal barrier is naturally more permeable, what some people call a leaky gut in adults.
So for them, BCM7 can be absorbed directly into the bloodstream in much greater amounts.
Yes, which allows it to potentially interact more directly with the central nervous system. And that raises a whole different set of concerns for early life development.
Let's transition to the immediate impact for most people. The gut issues. This is where we get into this idea of pseudo-lactose intolerance.
This is maybe the most crucial takeaway for anyone listening who struggles with dairy. The evidence strongly suggests that a huge number of people who think they're lactose intolerant, the bloating, the gas, the pain, are not actually reacting to the milk sugar.
They're reacting to the protein.
They're reacting to the physiological effects of A1 beta-casein and the BCM7 it releases. And these effects either perfectly mimic lactose intolerance or they make a very mild underlying intolerance feel much, much worse.
So how does an opioid peptide cause symptoms we normally associate with? Gas and fermentation.
It's all about motility. BCM7 activates those mu-opioid receptors in the gut's own nervous system, what we call the enteric nervous system.
The second brain.
The second brain, exactly. And activating those receptors is the body's natural brake pedal for digestion. So when BCM7 binds to them, it's a powerful signal to significantly slow down the whole gastrointestinal transit time.
So it's literally like taking an anti-diarrheal drug.
That's the perfect comparison. A drug like loperamide works by activating those exact same peripheral opioid receptors to stop motility. BCM7 does it naturally after you drink a glass of A1 milk.
And we have hard data on just how much it slows things down, don't we?
We do, and it's really compelling. There was a major human clinical study on Han Chinese adults who are genetically prone to have low lactase levels. They directly measured it.
And what did they find?
They found that the total time it took for food to pass through the digestive system was significantly longer, by an average of 6.3 hours, when they drank milk with A1 protein compared to when they drank pure A2 milk.
6 1/3 hours. That is a massive delay.
It's a huge delay. And that extra time is what causes the symptoms. It gives gut bacteria so much more time to ferment any unabsorbed sugars and proteins in the colon.
And that fermentation produces gas.
It produces gas methane, hydrogen. That's what leads directly to the bloating, the pressure, the pain that people blame on dairy intolerance.
Now, in the sources, there was a mention of a rapid emptying paradox. That seems contradictory. If BCM7 is a brake, how does it also cause rapid emptying?
The great point. It's about the difference between the stomach and the colon. In some trials, especially with people who already had some trouble digesting lactose, they saw that A1 milk actually emptied from the stomach more rapidly.
So it dumps into the small intestine faster.
Exactly. You get the sudden rush, this large load of undigested lactose hitting the small intestine all at once, which can cause intense pain and urgency. So you have this double whammy of rapid gastric emptying followed by an agonizingly slow transit through the rest of the system, a recipe for misery.
So BCM7 isn't just a physical brake, it's also an immune trigger. It's linked to inflammation.
Absolutely. The consumption of A1 beta-casein is strongly linked to heightened inflammation in the gut. And we can measure this. It's not just a feeling.
I didn't measure it.
We use established clinical biomarkers. For instance, in human trials, levels of fecal calprotectin, which is a protein released by immune cells and a gold standard marker for intestinal inflammation, were consistently and significantly higher after people ate an A1 diet compared to an A2 diet.
What does that kind of inflammatory response tell us about how the body sees this A1 protein?
It suggests the body is mounting a specific and undesirable immune response. You can see it in blood work too. A1 consumption is linked to increases in things like interleukin-4 or IL-4, which is a key player in allergic responses, and also higher levels of various antibodies like IgG and IgE.
So it looks more like an allergic reaction than a digestive issue.
The whole pattern points towards a Th2-mediated immune response, which is characteristic of the body reacting to something as if it were an allergen or a significant irritant.
And this inflammatory cascade has a secondary effect that ties everything back to this idea of pseudo-lactose intolerance.
This is the critical piece of the puzzle. The leading hypothesis is that this A1-induced inflammation directly downregulates the activity of the lactase enzyme itself.
So the inflammation damages the gut lining.
Precisely. If the lining of your small intestine is constantly inflamed from BCM7 and the resulting bacterial issues, the little microvilli on the surface, where the lactase enzyme is supposed to do its job, get damaged and become less functional.
Wait, so just to be clear, you're saying A1 doesn't just mimic intolerance? it can actually cause a secondary acquired lactose intolerance by damaging the gut environment.
That is the exact argument.
Yeah.
The root cause of the problem for many people isn't that they were born unable to produce lactase. It's that the A1 protein creates this hostile inflammatory environment that physically suppresses the lactase they do have.
That's a huge paradigm shift.
It is, and it provides the most elegant explanation for why so many people who are convinced they're severely lactose intolerant can suddenly drink a glass of pure A2 milk and feel perfectly fine.
Right.
They didn't magically regrow their lactase enzyme. They just removed the inflammatory thing that was shutting it down.
We have to start asking if dairy is a problem and start asking if A1 beta-casein is the problem.
It completely reframes the question from a sugar issue to a protein sensitivity.
Okay, let's follow that BCM7 trail out of the gut. It's a major player in the gut-brain axis. How exactly does this opioid peptide bridge that gap between our intestines and our central nervous system?
The gut-brain axis is this incredible two-way communication network. You have the vagus nerve talking directly, you have immune signals like cytokines, and you have metabolites from our gut microbes. BCM7 is a powerful actor on that stage.
Because it's an opioid.
Because it's an opioid, it has the chemical structure that could allow it to cross the blood-brain barrier. Now, in healthy adults, that barrier is pretty tight, but as we said, permeability can be increased, especially if the gut is already compromised or inflamed, and in infants, it crosses much more readily.
Do we have any concrete evidence that drinking A1 milk can actually impair brain function?
We do. We can look at the results from studies that use something called the Subtle Cognitive Impairment Test, or SCIT.
Okay.
These are tests designed to measure things like processing speed, attention, accuracy, the subtle mechanics of thinking. And the results were clear. Participants who drank conventional A1-containing milk showed significantly slower response times and made more errors on these tasks compared to when they were on A2 milk.
So a tangible slowing down of the brain's processing power.
Exactly. The implication is that the chronic metabolic stress, the inflammatory signals coming from the gut, and that opioid interaction, it all creates a kind of systemic drag. The body is diverting resources to deal with the gut distress, and the result is measurable brain fatigue.
And that would logically extend beyond just reaction speed to things like mood and psychological well-being.
It does. And that's where the newest research is heading. There's a study called the BIGS trial for brain immune gut skin that looks specifically at this.
And what did the BIGS trial find regarding mood?
They found that when people switched from conventional milk to A1-free A2 milk, they saw measurable reductions in their self-reported scores for anxiety, depression, stress, and fatigue.
That's significant.
It was statistically significant. Not a life-altering change, but a clear improvement in psychological well-being. And interestingly, the effect was even more pronounced in the female participants, which suggests there might be some sex-specific interactions going on.
What's the proposed mechanism there? How does drinking A2 milk make you feel less anxious?
Well, getting rid of the BCM7 is part of it, but it's broader than that. It seems to come down to the microbiome. Switching to A2 milk appears to create a healthier gut environment, which then changes the profile of short-chain fatty acids, or SCFAs, that the microbes produce.
And those are key signaling molecules.
They're one of the main ways the gut talks to the brain. SCFAs, especially one called butyrate, are critical for gut barrier health, reducing inflammation, and they even influence neuroinflammation in the brain itself. A healthier gut just sends better, less stressful signals upstairs.
This brings us to the most complex and historically the most controversial area: long-term epidemiological risks. And we should be very clear here, regulatory bodies have been cautious.
That's the essential context, yes. Back in 2009, the European Food Safety Authority looked at the data and said, look, a definitive cause and effect link between A1 milk and chronic diseases has not been proven for public health. But
the research into strong correlations and plausible mechanisms has continued.
Exactly. We're investigating very plausible biochemical pathways, even if we don't have definitive proof of causality across the whole population.
So let's start with type 1 diabetes, T1D. It's an autoimmune disease.
Correct. The body's immune system attacks its own insulin-producing cells in the pancreas. And epidemiologically, for decades, researchers have noted a very strong global correlation between how much A1 milk a country consumes per capita and its incidence of T1D.
And you see lower rates in places that consume more A2 milk.
Right. Places like Iceland, which historically had very low A1 exposure, or Japan, where traditional dairy wasn't a big part of the diet, they have much lower rates of T1D.
So what's the hypothesis? How could BCM7 trigger an autoimmune attack on the pancreas?
The primary theory is called molecular mimicry. The idea is that the BCM7 peptide, or a fragment of the A1 protein that releases it, looks chemically very similar to a sequence on our own pancreatic cells, specifically on the GLUT2 glucose transporters.
So the immune system gets confused, it sees the milk protein, thinks it's a foreign invader, and makes antibodies against it.
Exactly. But because of this structural similarity, this molecular mimicry, those antibodies don't just attack the milk protein fragment, they also attack the body's own GLUT2 transporters on the pancreas.
And those transporters are vital.
They're vital for the pancreatic beta cells to sense blood sugar and release insulin properly. So by attacking them, the immune system effectively targets and destroys its own insulin-producing machinery, which is the direct cause of type 1 diabetes.
A complex process. Okay, let's move on to cardiovascular health. Atherosclerosis, coronary heart disease, what's the proposed link there?
Here the focus shifts to BCM7's effect on oxidative stress and the lining of our blood vessels. Most of the evidence for this comes from animal models, specifically with rabbits.
What did they find in those studies?
They found that rabbits fed a diet with A1 beta-casein developed significantly more fatty streaks and early lesions in their aorta compared to rabbits fed A2 beta-casein. And fatty streaks are the very first, earliest stage of atherosclerotic plaques.
Okay, so we need to connect the dots. What's the biochemical cascade that gets us from BCM7 to those fatty streaks?
It's an indirect but really powerful mechanism. It revolves around the body's master antioxidant, glutathione or GSH. To make glutathione, our cells need a key amino acid called cysteine. It's the rate-limiting step. And research has shown that BCM7 can inhibit the ability of our cells to take up cysteine.
So if BCM7 is present, our cells can't get enough cysteine.
And if they can't get enough cysteine, they can't produce enough of the master antioxidant, glutathione.
Which leaves the whole system vulnerable.
It leaves your vascular system highly vulnerable to oxidative stress from things called reactive oxygen species. This oxidative stress damages cell membranes and, most importantly, it promotes the oxidation of LDL cholesterol. And oxidized LDL is the absolute definitive starting point for the formation of those atherosclerotic plaques.
So again, it's this incredibly deep cascade. A single amino acid change leads to an opioid peptide, which leads to antioxidant depletion, which then increases heart disease risk.
It really highlights how interconnected everything is when it comes to nutrition and chronic disease.
Now, you also mentioned some more speculative neurological links. SIDs, autism, schizophrenia, we should probably touch on those just to be thorough, but with a big dose of scientific caution.
Absolutely. We have to stress these are hypotheses. They lack large-scale human clinical data. The first one, relating to sudden infant death syndrome, or SIDs, revolves around BCM7's opioid activity.
Okay.
The theory, which has some support from mouse studies, is that if an infant with an immature blood-brain barrier absorbs a significant amount of BCM7, the opioid effect could reach the central nervous system and suppress the respiratory drive. It could cause apnea, a pause in breathing.
A very serious theory, even if it's unproven in humans. What about the link to developmental disorders?
That's rooted in the leaky gut idea again. The theory proposes that in individuals with a highly permeable gut, especially during critical periods of neurological development in childhood, an excess of absorbed BCM7 might interfere with neurotransmitter balance and brain development.
Have there been any studies on that?
There have been some small clinical reports, anecdotal evidence of symptom improvement when A1 is removed from the diet. But honestly, until we have large placebo-controlled trials, the scientific consensus has to remain very, very cautious about that link.
Okay. This whole discussion paints a really compelling picture. Let's switch gears a bit to the practical side of things. Does this A1-A2 difference actually change the physical properties of milk itself? Does it matter for, say, a cheesemaker?
It absolutely does, profoundly. That single amino acid change alters the structure of the casein micelles, which are the tiny protein structures that carry calcium in milk. A2 beta-casein results in micelles that are generally a bit larger and have a lower negative charge compared to A1.
And how does that play out in an industrial setting, like making cheese or yogurt?
It mainly affects coagulation, how the milk sets. A2 milk generally has poorer coagulating properties. It takes longer to set, and the curd it forms is looser, less firm.
So for a cheesemaker, that could mean lower yields.
That's exactly what studies have reported. You can get lower cheese yields from A2 milk, because the protein matrix is less dense, so it doesn't trap as much fat and moisture. So from a purely industrial efficiency standpoint,
A1 milk might actually be better for making certain cheeses.
in some ways, yes, it gives you a tighter, faster setting curd. But here's the paradox for the consumer. While A2 milk is slower to set in the vat, the gels it forms, like in yogurt, tend to be softer with larger pores.
And that might actually make it easier to digest.
Ironically, yes. That softer structure might facilitate more rapid and gentler digestion in the human stomach compared to the tighter gels from A1 milk.
That's fascinating. So a trade-off. Slower for the factory, maybe easier on the person.
Exactly. And speaking of processing, we have to talk about pasteurization. This affects almost all the milk we drink.
Does the heat treatment make that weak link in A1 milk even weaker?
It certainly appears to. There was a really important Polish study back in 2007 that looked at this. They found that while you can detect tiny trace levels of BCM7 in raw A1 and A2 milk, the concentration of BCM7 in the A1 milk surged dramatically after it was put through high-heat pasteurization.
So the very process that makes milk safe could be inadvertently maximizing the release of this problem peptide from A1 milk.
That is the critical implication. The heat stress seems to destabilize that weak histidine link even further, making it even easier for our enzymes to cleave it later. It might help explain why some people report doing better on raw milk, which often comes from smaller, heritage A2 dominant herds anyway.
It's a double effectless A1 to begin with, and no heat to make it worse.
A very important combination.
Now, we've focused so much on the beta-casein itself, but the breeds that produce A2 milk Jerseys, Guernseys, they're just different cows. They have a different nutritional profile than Holsteins, don't they?
It's a huge factor that often gets overlooked. When you switch to an A2 brand, you're often getting a nutritional upgrade just because of the breed. You really have to compare the milk from a Jersey to the milk from a Holstein.
Let's run through those numbers because they're pretty significant.
They are. Jersey milk consistently has about 19 to 21% higher total protein content. And even more dramatically, it has about 36% more calcium. We're talking 1.5 grams per kilogram versus about 1.1 in Holstein milk.
And what about the fat content? I know a lot of people see that as a negative, but it's important.
It's incredibly important for nutrient density and flavor. Jersey milk is naturally higher in fat, around 5% compared to maybe 3.5% for Holsteins. That means more energy, more fat-soluble vitamins, and a higher proportion of those beneficial short-chain fatty acids we talked about earlier.
Before we wrap up the nutritional side, what about trace minerals? Does farming practice play a role there?
It's a key caveat, yes. Farming style can sometimes override the breed genetics. Interestingly, some studies have found that organic milk, which often happens to be A2 dominant because of the breeds they use,
Okay.
sometimes lower in essential minerals like iodine and selenium compared to conventionally produced milk, just because of differences in the feed and soil.
So if you're making the switch to A2 for health reasons, you might need to be mindful of getting those specific minerals elsewhere in your diet.
Precisely. The A1, A2 choice is a biochemical one, but the overall nutrition is still tied to how that cow was raised.
Finally, let's talk about the commercial reality. If you're listening and you think you might have an A1 sensitivity, can you actually find A2 milk? And what's the cost? We looked at Kailua, Hawaii as an example of a high-cost market.
Right. And even in a competitive high-cost place like that, A2 milk is completely mainstream now. Brands like A2 Milk or local lines like Alexandre are in all the major retailers, Safeway, Whole Foods, you name it. It's not a niche product anymore.
But that accessibility comes with a premium price tag, doesn't it?
It absolutely does. You should expect to pay a significant premium. If a conventional half gallon is, say, $4, the A2 brands are probably going to be somewhere between $4.50 and even up to $10 for a specialty organic A2 product.
And that price reflects the cost of genetic testing, smaller herds.
And often, that superior nutritional density from the heritage breeds. It's a premium that clearly a growing part of the market is willing to pay.
Okay, we've gone from a single gene mutation 8,000 years ago to the price on the shelf today. Let's try to synthesize all of this. What are the cumulative advantages of A2 milk for our listener?
I think we can boil it down to 4 key areas where A2 offers a really critical advantage over conventional milk, especially for anyone who's sensitive.
Let's start with number 1, digestibility.
Right. A2 milk prevents the release of BCM7. That means it avoids that opioid-driven slowing of the gut, that 6.3-hour delay we talked about. This reduces fermentation, gas, and discomfort. For many people, this alone is life-changing.
Number 2, Inflammation.
It promotes a healthier gut environment. A2 milk lacks those pro-inflammatory triggers that raise markers like fecal calprotectin. By keeping inflammation down, it helps protect the function of your natural lactase enzyme, preventing that secondary intolerance.
Number 3, the brain and cognitive function.
The switch away from A1 seems to protect your central processing. By reducing inflammation and avoiding BCM7, it protects cognitive speed and accuracy, as we saw in the SCIT trials. And the changes to the microbiome may support better mood outcomes.
And finally, number 4, nutrient density.
Because it so often comes from those heritage Jersey and Guernsey breeds, you're usually getting a nutritionally superior product. More protein, significantly more calcium, and more energy-dense beneficial fats compared to the Holstein standard.
So for anyone out there with these undiagnosed dairy issues,
A2 milk represents a really well-substantiated, promising alternative. It helps avoid the immediate digestive problems and the pathways that are linked to potential long-term risks. The clinical data on the immediate effects is strong enough now to make this a mainstream recommendation for sensitive people.
that brings us to our final thought. The science on the short-term benefits is getting clearer and clearer, but the long-term causality is still the big question mark.
It absolutely is. The next critical frontier for research has to go deeper than just BCM7. We need long-term studies that look at the precise interaction between these A1 and A2 proteins and the entire gut microbiome.
So beyond just general changes.
Way beyond, it raises an important question. What specific sustained microbial shifts happen in the gut when it's constantly exposed to A1 versus A2? And how do those long-term shifts in our gut bacteria either compound or maybe even mitigate the effects of BCM7 exposure over a lifetime? That's the research that will finally settle the debate on chronic disease.
A fascinating area to watch. This has been a truly rigorous deep dive into a product we all thought was so simple. Thank you for walking us through the incredible complexity of all this.
My pleasure. It was great.
And thank you for joining us on the deep dive. We hope this has given you a whole new, much more granular perspective on what's in your glass.